Okay, so welcome to this next video in which we are discussing the glucocorticoids for the treatment of asthma. Okay, so we're now going to discuss the pathology of asthma, basically, so that we can understand how glucocorticoids can be used to treat asthma. Okay, so we're going to start off with what happens when you undergo your primary exposure to the allergen. Okay, so this will happen years and years ago, basically, when you were potentially a toddler. Okay, so you're going to inhale the allergen for the first time, and this is called primary exposure. So the first ever time you're exposed to the allergen. Okay, so what's going to happen is the allergen is going to be breathed in, okay? It's going to go into all of the airways, and some of it will end up within the lamina propria of the airways, okay? So some will just happen to get past the epithelial cells, and some allergen will end up in the lamina propria. Now, in the lamina propria, there are two types of cell which are capable of phagocytosing this allergen and then presenting it to T cells in order to activate what's known as a humoral adaptive immune response. So there are two main strands of the adaptive immune response. There is a humoral adaptive immune response and a cell-mediated adaptive immune response. When the pathogen is extracellular, you launch an humoral adaptive immune response, whereas when it's intracellular, you launch a cell-mediated adaptive immune response. In this case, the allergen is extracellular, so this is going to count as an extracellular pathogen. So we're going to launch an a humoral adaptive immune response. Okay, right. So what's going to happen? Well, the allergen is going to be phagocytosed by these two professional antigen-presenting cells. Now, what are the two professional antigen-presenting cells? Well, you have two. One is known as a dendritic cell, so let me show you this. Okay, so dendritic cells have these dendritic processes coming off their cell body here. Now, they have absolutely nothing to do with neurons, even though they look like the cell body of a neuron, okay? They are not capable of conducting action potentials. They're not excitable. They simply do not have the voltage-gated ion channels that you need in order to conduct an action potential. Okay, instead, they are a professional antigen-presenting cell, okay, which means that they are capable of activating T cells. Okay, so this is a dendritic cell. Now, you have dendritic cells sort of interdispersed among all of the tissues of your body, so you have them dotted around everywhere. Okay, another type of professional antigen-presenting cell is what's known as a resident macrophage. So let me show this here. Okay, so this represents a resident macrophage. Okay, so again, interdispersed amongst the cells of any tissue within your body, you will have resident macrophages, and these are cells that are just sitting around and waiting for pathogens to arrive so that they can launch an adaptive immune response against them if necessary. Okay, so in comes our allergen. Okay, so here is our allergen protein. Okay, so all allergens are proteins because you can't launch a humoral adaptive immune response against something that's non-protein. Okay, so currently this is the entire protein. Now what's going to happen is this protein is going to be engulfed into a phagosome, okay, within our professional antigen-presenting cell, and I should write that down. That's the good key term. Okay, so professional antigen-presenting cell. Now, often people drop the professional. To emphasize it, you should say professional antigen-presenting cell, because in actual fact, all cells of the body can present antigen, and, well, what I've said is slightly wrong. All nucleated cells of the body can present antigen. Erythrocytes can't present antigen, for instance, because they don't have a nucleus. But all nucleated cells can present antigen, so calling these antigen-presenting cells is a little bit unfair on all the other cells of the body. So to emphasize that, often, well, occasionally, people write professional antigen presenting cells to be absolutely proper, but everyone just calls them antigen presenting cells. So when people refer to an antigen presenting cell, you can safely assume they mean a dendritic cell or a resident macrophage, and they don't mean just any old nucleated cell. Okay, because any old nucleated cell is not capable of activating T cells. Okay, well, they can be capable of activating memory T cells, but not naive T cells. Okay, so these are special in that they can activate naive T cells. Okay, so T cells which have never seen their antigen before. Okay, and remember, we've never seen this antigen before. This is the first time you've ever breathed it in. So maybe it's your first year of life, and the trees have blossomed for the first time, and you're breathing in this pollen for the first time. Okay, so what's going to happen is the allergen is going to go into a phagosome within the professional antigen presenting cell here. Okay, and I'm going to use the resident macrophage throughout this entire discussion, simply because it's so much less effort to draw. The dendritic cell is very, very difficult to draw. It requires effort. Okay, this one is far easier to draw, and it's far easier to draw later when we've got to draw immune synapses. It's a nightmare to draw immune synapses on a dendritic cell, whereas it's quite easy to draw them on a resident macrophage. Now, this is kind of unfair, however, because dendritic cells are actually far better at presenting antigen to naive T cells than resident macrophages are. Resident macrophages can do it, but it's dendritic cells that usually initiate adaptive immune responses. Okay, but as I say, just for ease of drawing, we'll draw a resident macrophage. Okay, so it's phagocytosed the allergen, and now what's going to happen is that it's going to break this allergen down into fragments, okay? And these fragments are going to be put on the surface of the resident macrophage, and they're going to be bound to MHC class II. So whenever you have a protein that you got from the external world, so these professional antigen presenting cells, the way they got this protein was they phagocytosed it up, they engulfed it into this phagosome here. Whenever you've got a protein that you got from the external world rather than from your own cytoplasm, and you've chopped it up and you're going to present it on your surface, you will present it on major histocompatibility complex class II. Okay, so this is a complex of a whole bunch of subunits, okay, called the major histocompatibility, and this is the worst word we have to use, I think, in this entire video. Histocompatibility complex, there, it just fitted in, complex, and then it's class II. And from now on, I will not rewrite out the major histocompatibility complex class II. Instead, I will denote it the way everyone denotes it, which is MHC, major histocompatibility complex, and then class II on the end. Okay, so here is our major histocompatibility complex class II complex of proteins on the surface of the professional antigen presenting cell, and what we're going to do is we're going to stick fragments from our allergen on the surface of that MHC class II. So we're going to stick a little fragment of the allergen in here. Okay, now of course you will get a huge number of different fragments from this allergen. Okay, so we will follow the story of just one of these fragments, but loads of them, loads of different fragments will be put on the surface of this professional antigen presenting cell in the context of MHC class II. Okay, now what's going to happen is our professional antigen presenting cell is going to go into lymph vessels, okay? So the lamina propria, as well as having blood vessels in it, will also have lymphatic vessels, okay? So something a little bit more fancy. But of course all tissues have them, we forget it, but all tissues do have lymphatic vessels. Okay, so here is a lymphatic vessel, or a lymph vessel. So these macrophages are going, or dendritic cells, this professional antigen presenting cell that has this antigen fragment here, okay, and I will refer to it as an antigen, because it is an antigen. As I said, all allergens are antigens. It's just an antigen that doesn't deserve an immune response being initiated. And I'm sorry, I couldn't fit in the word fragment, so this should say antigen fragment. And by the way, the abbreviation for antigen is AG, for antigen. Okay. So, the professional antigen-presenting cell is going to go into the lymph vessels which drain the lamina propria, and it's going to pass along to a lymph node, to a local lymph node. Okay, so loads of afferent lymph vessels come into one lymph node. So let me show you the structure of a lymph node, because I always think lymph nodes have quite an interesting structure. They're very well suited to what they need to do. Okay, so here are the afferent lymph vessels. Okay, and then coming out of the lymph node is then a big efferent lymph vessel. So all of these five, these small ones, these are what are known as afferent lymph vessels. Okay, whereas this one big vessel coming out of the lymph node, draining the lymph out, is an efferent lymph vessel. So efferent means it's going away from, afferent means it's coming into. Okay, so afferent lymph vessels bring lymph and professional antigen-presenting cells into the lymph node. Efferent lymph vessels take it out. Okay, then what happens is the lymph first arrives in something known as the subcapsular sinus. So all around the outside there is a space that is absolutely full of lymph fluid, basically. Okay, and this space around the edge of the lymph node is what's known as the subcapsular sinus. Okay, so it's just underneath the capsule, which is why it's the subcapsular, and sinus just means a fluid-filled space. Okay, then all this lymph is coming in from all these different afferent lymph vessels, and now what has to happen is it has to somehow get to this efferent lymph vessel over here, and to do that it has to drip or dribble or percolate is the fancy word, through a whole bunch of T cells, a whole bunch of cells that are sort of scanning this stuff that's coming through them and just checking to see whether we can launch an immune response against any of it. And that's the purpose of these lymph nodes. They are immune surveillance. They are looking at all the lymph that is coming in from all these afferent lymph vessels, and they are saying, is there anything dangerous here? Should we launch an adaptive immune response against any of it? That's what lymph nodes are doing. Okay, right. So, lymph nodes have two layers. Okay, one layer is known as the cortex. So we'll say this is the cortex, and the cortex is absolutely full of T lymphocytes. Okay, naive T lymphocytes, and also some memory T cells, but we'll talk about those later. For now, we're just interested in the naive T lymphocytes, and certainly the naive T lymphocytes will be the bigger population. Okay, so all over the place, basically, you have T lymphocytes. Now, dotted around in the cortex, to make it a bit more interesting, you have spheres or follicles, which aren't T lymphocytes at all. Instead, these are spheres of absolutely loads of B lymphocytes that can't stand to be amongst T lymphocytes, and therefore all gather together into a great sphere to cluster together, basically. Okay, so these spheres here, these are spheres made up of B lymphocytes, absolutely loads of B lymphocytes. So it's just a sphere of B lymphocytes, basically. Okay, and these are in the cortex of the lymph node. So they are what are known as the B cell follicles within the cortex. Okay, so B cell follicles. Okay, now the rest of the cortex around the B cell follicles, which I'll highlight in green here, this is the T cells. Sorry, not in green, I've highlighted it in blue. This is the T cells, okay? So I might even colour it in properly rather than this sort of cheap dashing. Okay, so here are these T cells in blue here, and they sort of make up the rest of the cortex. So the B cell follicles are just sort of dotted in amongst the T cells that make up most of the cortex. Okay, and what's going to happen is that the lymph, in order to get from the subcapsular sinus to that efferent lymph vessel, is going to have to come through all of these T and B cells, okay? And they are going to be scanning it continuously, looking for things that we can launch an adaptive immune response against. Okay, and then you have another layer of T cells, known as the paracortex, which has these little protrusions into this final sinus here. So this middle sinus here is known as the medullary sinus. Medullary means middle. Okay, so this is the sinus in the middle of the lymph node. Okay, and it leads on to the efferent lymph vessel. So lymph is trying to get from the from the subcapsular sinus into the medullary sinus, and to do that it has to firstly pass through the cortex, which is made up of loads and loads of T lymphocytes with these B cell follicles amongst them. And then it also has to go through this what's known as a paracortex, the second layer of T cells. And here there are no B cell follicles amongst the T cells, but there are again loads and loads of T cells. So this is the paracortex here. So I always quite like that. I like the way the lymph has no choice but to percolate amongst all of these lymph nodes. Okay, right. So what's going to happen is that our professional antigen-presenting cell is going to come up this afferent lymph vessel. It's going to go into the subcapsular sinus, and it's then going to percolate through the cortex and the paracortex. Now, it's trying to activate a T cell. Okay, now, before we can go any further, we need to discuss the two separate populations of T cells. So there are two separate populations of T cells. So let me draw these T cells here. Now, T cells are very easy, well, in fact, lymphocytes are very easy to distinguish because they have absolutely massive nuclei compared to their cytoplasm. So they have very, very little cytoplasm. And the reason is that this T cell sits and does nothing all day. Okay, so it sits in these, the cortex and the paracortex of lymph nodes. Now, it's got a slightly more interesting life than just sitting in the same lymph node forever. It can move out of that lymph node into the blood and then go to another lymph node, or potentially go to the spleen. The spleen also has T cells and B cells within it in the white pulp of the spleen. Okay, so it can move around a bit, but it's not doing any chemical reactions, not any strenuous chemical reactions. I mean, it will be, obviously, it will have a tick over rate of respiration, but compared to other cells which might, you know, be producing a huge number of proteins, its metabolic output is rather feeble, so it doesn't need much cytoplasm because, as you probably learned in year 7, the cytoplasm is where all the chemical reactions occur. If it's not doing that much chemical reactions, then it doesn't need a very big cytoplasm. Okay, so there are two main types of T cell. Okay, naive T cell is what we're talking about now. So the types of T cell that are mainly within the cortex and the paracortex are naive T cells. There will be some memory T cells, but we'll come to those later. Okay, so there are two distinct populations of naive T cells. There are CD4 positive naive T cells, and there are CD8 positive naive T cells. CD4 positive naive T cells have CD4, a protein on their surface in the membrane, basically. Okay, so let's say this is CD4. CD8 positive naive T cells do not have CD4 on their surface, and instead have CD8. So no naive T cell has both CD4 and CD8. In the developmental stages, they can have both CD4 and CD8, but these are mature now, mature naive T cells, okay? So they do not have both of them. They will have one or the other, okay? So loads of these naive T cells in the cortex and the paracortex will have CD4 on their surface, and loads of them will have CD8 on their surface. And I shouldn't have put a positive there. The molecule is known as CD8, which stands for cluster of differentiation 8, or cluster of differentiation 4. Okay, so we are not interested in these CD8 positive naive T cells at all. they are involved in the cell-mediated response. In order to activate a CD8 positive naive T cell, you need to present your antigen fragment on MHC class 1. We are presenting it on MHC class 2. If you present it on MHC class 2, you can activate CD4 positive naive T cells. Okay, so we've immediately eliminated a huge great population of T cells for activation. We can't activate the CD8 positive naive T cells with this professional antigen presenting cell that's presenting this allergen fragment on MHC class 2. Okay, so it has to activate these CD4 positive naive T cells. Now, does it go in and activate absolutely every naive CD4 positive T cell? Well, of course, no is the answer to that. Basically, even in this population of CD4 positive naive T cells, there is variation. So not every naive CD4 positive T cell is the same. Instead, every naive CD4 positive T cell has its own T cell receptor. So let's say this is our naive CD4 positive T cell. Okay, so if you look at one naive CD4 positive T cell and then look at another one, you'll find that they differ in a certain protein on their surface. Okay, and this protein is again a protein complex. It's made up of a whole bunch of proteins, but of course you can consider that a protein if you want by the quaternary structure of proteins. Okay, it's known as the T cell receptor. Okay, so this is the T cell receptor, and T cell receptor is often abbreviated just to TCR. Okay, now the basic structure of the TCR will be the same amongst all different naive CD4 positive T cells. However, this little portion here that I've drawn just as a little sort of furrow, this is the region that's going to bind and recognize a certain antigen fragment that's being presented by a professional antigen presenting cell, and this portion will differ for all the different naive CD4 positive T cells. So, this massive great population of CD4 positive naive T cells that is within the cortex and the paracortex of the lymph node, it's got a massive variation within it, basically. All of the different members of this population have a different T cell receptor which recognizes a different allergen. Okay, so what has to happen is this professional antigen presenting cell has to find a naive CD4 positive T cell that has a perfect T cell receptor for binding to the allergen fragment that it has got presented on MHC class 2. Okay, so here comes our antigen presenting cell here, and remember I said that I would often just abbreviate antigen presenting cell to APC. Here is its MHC class 2 here, and then bound to the MHC class 2 it's got this allergen fragment, and let's say it just happened to find a naive CD4 positive T cell that had a T cell receptor that was complementary to this allergen fragment. Now you might say, surely the probability of that happening is, you know, humongously low, but you have to remember that firstly each antigen presenting cell that chopped up the allergen would get many different fragments of that allergen, and it will have them all on MHC class 2 molecules. So actually it doesn't really care which allergen fragment it's going to actually use. It just needs to find a T cell that's got a receptor that's complementary to one of them. Okay, so that increases the probability it will find a T cell that's complementary to one of them. Okay, in addition, you have to remember that it's not just one antigen presenting cell that is doing this task. You're sending absolutely loads of antigen presenting cells to the lymph nodes. Then they've all got this allergen, they've all got fragments of the allergen chopped up on their surface, so they're all searching for naive CD4 positive T cells which have a T cell receptor which will bind to one of these fragments. Okay, so that increases the probability that eventually you will find a naive CD4 positive T cell which has a T cell receptor which is complementary to one of the allergen fragments which we got from this allergen. Okay, so let's colour in the allergen fragment again. So it's in turquoise here. So to make the story simple, we will continue imagining that this little allergen fragment that we specifically coloured in turquoise here happened to find a CD4 positive naive T cell which has the T cell receptor that's complementary to it. Okay, so to keep it nice and simple, we won't draw all the different allergen fragments and note the possibility that you could launch many different types of CD4 positive T cell off, which have T cell receptors that are launched against totally different allergen fragments, which all came from the same allergen. To keep it simple, we'll just have this one allergen fragment on this MHC class 2 molecule. Okay, so here is the MHC class 2 in red here. Okay, and it's bound to that T cell receptor on the surface of the CD4 positive naive T cell. Okay, so this is MHC class 2, and this is the T cell receptor. Now, what colour should I colour in the T cell receptor? I think I'll colour it in orange. Right, now, we need to understand why it was so important that this T cell had CD4 on its surface. Well, basically, the T cell receptor is going to bind to the allergen fragment as well as the MHC class 2, and that's an important thing to stress. It doesn't just bind to the allergen fragment, it does bind to the MHC class 2 as well, okay? But importantly, CD4 also binds to MHC class 2, and it only binds to MHC class 2. It doesn't bind to MHC class 1, whereas CD8 on these T cells over here binds to MHC class 1, and that's why you need to present antigen fragments on MHC class 1 if you want to activate CD8 positive naive T cells. Okay, so we're presenting it in MHC class 2, so we are going to bind to a CD4 positive naive T cell because we're going to need CD4 here. Okay, so now what's going to happen is once the T cell receptor is bound to the allergen fragment on the MHC class 2, and the CD4 has also bound to the MHC class 2, this is going to send what's known as signal 1 to the naive CD4 positive T cell. So the naive CD4 positive T cell now receives signal 1 for activation. Okay, now signal 1 alone is not, repeat, is not good enough to activate this naive CD4 positive T cell. Instead, to activate the naive CD4 positive T cell, the antigen presenting cell needs to have co-stimulatory molecules on its surface which can activate receptors on the surface of the naive CD4 positive T cell. Okay, so it's going to have two major molecules on its surface, okay? So let me draw these here. So you're going to produce CD8, the cluster of differentiation, CD40, the cluster of differentiation 40, and you're going to put that on your membrane. You're also going to produce B7.1 or B7.2, and this is also going to be very, very important. Okay, so these are the co-stimulatory molecules. So B7.1 or B7.2, and both of these two molecules also have other names. So B7.1 is also known as CD80, and B7.2 is also known as CD86. Okay, now what actually induces the expression of these co-stimulatory molecules, and I should write that because it's so important somewhere. I'll put it up here because I want to leave that space free. So you're going to put these co-stimulatory molecules on the surface of the antigen-presenting cell. Now, what actually induces the expression of these co-stimulatory molecules on the surface of the antigen-presenting cell? Well, basically, you need activation of the pattern recognition receptors on the antigen-presenting cell. Okay, so let's say this is a pattern recognition receptor here. Okay, basically, pattern recognition receptors bind to molecules that are associated with pathogens, and these are broad molecules that that human cells would never ever have on their surface, which pathogens can have on their surface. Okay, and we'll continue this discussion in the next video.